This video is about how we would create a transgenic plant. So it's very similar to bacterial transformation. In fact, it involves bacterial transformation. And then what we do is we actually use the bacteria to do what they're good at, which is infecting other organisms. So these are a very cool process to see how we can. So manipulating a bacterium is quite easy. Their genome is quite simple. Manipulating a plant is a lot more difficult. Their genome is like our genome. Very complex, lots of different genes. They don't have plasmids. So how can we do that? We go through this process. So now we're doing this second part here, their use in agriculture. Okay, so we're going to talk about how we use these in agriculture. So we're still on page 6 in your notes, and there are some gaps for us to fill in. So like we talked about earlier, we isolate a gene. Yes, so whatever the gene might be. So maybe we gene isolate something for pest resistance or herbicide resistance. It doesn't matter which one. So pest resistance, and then we amplify it using PCR. So we get lots of copies. We then insert that gene into a vector plasmid and it becomes a recombinant plasmid. Yeah, so this is the same process as bacterial transformation. And then we insert that all into a bacterium. In this case, the bacterium is Agrobacterium tumefaciens. Now, this is the very cool bit. This is the bit that is different. So the first stage is the same. It's the same as bacterial transformation. Take a gene out of one thing, put it into a plasmid, put it into a bacterium. But instead of growing the bacterium now, what we actually do is we get the bacterium next to some baby plant cells. Okay, so these are plant cells, like, for example, the tobacco plant. I know we don't really want to talk about a tobacco plant, but they're quite good at doing this process. And what happens is the bacteria actually infect the plant and insert the gene. It's like they act in a similar way to a virus. So they are putting their genes in those plasmids into the plant cell. That's pretty cool. So after they've done that, we grow these cells in a lab. So this is them being grown. This is number 6 up here. And that means that all these little plantlets past here, they all have the gene that you inserted. So you start off by changing a baby plant or just a couple of plant cells, and then you grow a whole plant out of that. And most, if not all, of the cells of that plant, because they are clones or down the same cell line, they will have the gene of interest. And so now the whole plant, even though it's a complex organism, is transgenic and has the DNA to code for whatever characteristic you gave it. And that's in the next video, is talking about the different characteristics we can give to plants.